Our next speaker is Dr. Patrick Reynolds. He grew up in El Paso, Texas, received his BA in biology from the University of Texas at Austin, his MD from University Southwestern Medical School in Dallas, his PhD in cell biology from University of Texas in Austin, and his pediatrics training at the National Naval Medical Center. His postdoctoral fellowship was in cancer immunology at UT Southwestern. He holds the rank of commander in the U.S. Navy, retired now. He is currently the Cancer Center director for the School of Medicine at Texas Tech University in Lubbock. Dr. Reynolds' research focuses on cancer developmental therapeutics, and he pioneered the development of the differentiating agent cisretinoic acid for treating the childhood cancer neuroblastoma. His most recent research focuses on two areas: use of telomere maintenance mechanisms as cancer biomarkers for neuroblastoma and other cancers, and developing agents to specifically target alternate telomere lengthening cancers. He's also assessing GD2 expression on neuroblastoma cells as a mechanism of resistance to chemoimmunotherapy and developing novel non-GD2 anti-neuroblastoma antibodies. His other interests include mechanisms of resistance to drugs and antibody therapy in childhood cancer and adult cancers, and establishing and biobanking of cancer patient-derived models. He's a co-author of hundreds and hundreds of papers and peer-reviewed articles, and his talk today is going to be on GD2 expression as a biomarker for the treatment of neuroblastoma. Dr. Patrick Reynolds.
Thanks, Pat. Thanks for the introduction and for the invitation here. And thank you for your dedication to keeping this meeting going. It's a pleasure to be with you all. The reason that those of us who work on neuroblastoma is all of you. You're why we do what we do. Is this control working? Oh, there's one right here. Okay, great. Thanks. All right, so there's my conflict of interest statement, and there we go. So what we're going to talk to you is you've heard about GD2 both as an antibody therapy, and you recently heard about the elegant work on that not only the laboratory in Rome but others are doing, trying to develop CAR T cells. And I find it interesting because Carl June, who pioneered CAR T cells in leukemia, and I were in the Navy together, so that's another long story. But the GD2 antigen that's on that on neuroblastomas that's targeted by any of those kinds of immunotherapies is a very complex molecule, and we're going to talk to you a little bit about this today. As you might imagine, it's found in large amounts on the neuroblastoma cells, and that's why we're using it to try and target it with immunotherapies. But multiple different antibodies to GD2 exist, okay, but they all target GD2. All right, so in the end, they're targeting the same single molecule. So keep that in mind. And as you've heard, GD2 is being investigated for other approaches targeting drugs attached to antibodies and CAR T cells. What we're going to talk about today is really focusing on analyzing the amount of GD2 is on there and the potential for this as a biomarker for resistance to these kinds of immunotherapies. Now, we're going to focus mainly talking about the ADCC, or antibody-dependent cellular cytotoxicity mechanism of using antibodies. And this is applicable to any of the other. There we go. But this particular approach is really how dinutuximab beta, the other antibodies work, okay? And what happens is they target our regular immune cells, and I'm going to focus on neutrophils because that's my favorite cell because there's more neutrophils than anything else. But of course it could be monocytes or natural killer cells that the antibody binds with its back end to what's called an Fc receptor and basically arms these cells so that they will go out and kill a cell that the antibody can bind to. then the antibody's got to bind to the antigen. In this case, it's GD2. So the GD2 is represented there by these little blue triangles, and that's what the antibody's binding to. And once a neutrophil or any of these other cells is attached to the tumor cell, then it's triggered to kill that cell, and that's how this works. However, if you don't have GD2, or if you've lost GD2 on the tumor cell surface, as illustrated here, the problem is basically that cell's not going to attach to the tumor cell, and it's not going to kill it. So that's the simple mechanism here of resistance we're talking about. You have to have that complex molecule on the surface of the neuroblastoma cells in order to get the cell killed. Now, here's your biochemistry lesson for the day. These are the 9 synthetic steps necessary to make GD2. If you're targeting a protein, there's one step. You know, the RNA is made into protein, and the protein goes out on the surface of the cell. But GD2 is this complex lipid plus sugar molecule that's stuck together by these various 9 steps. For example, taking a fatty acid, palmitoyl-CoA, and conjugating it with an amino acid, that's the very first step. But you go through all these, part of it's the ceramide synthesis pathway, but you have to get through all of that to get to the end through these 9 steps to get GD2, and then it's got to get on the cell surface. And the point being here is that each one of those steps means there's a gene that's making that enzyme that makes that step work. So there's 9 different genes that could be mutated or lost in the process of making GD2, and therefore there are 9 opportunities to lose the synthesis of this molecule as we treat cancer cells. Now, to refresh your memory about what you learned in high school biology about selective pressure, illustrated here is this group of mice that has a mixture of black and tan mice, and then the hawk comes into the area, and you get a bunch of hawks, and pretty soon what you end up with is virtually all black mice, because the tan mice are easily targeted by the hawk. The hawk sees the tan mice, and he gets them. So the selective pressure is that tan mice don't survive. Black mice survive, they proliferate, and they take over the population. Simple selective pressure. Same thing happens with cancer cells. But before we get to that, let's remind you that when you give chemotherapy to any patient, including a neuroblastoma patient, these DNA-damaging chemotherapy agents are highly mutagenic. Okay? They're going to cause DNA damage, as is illustrated here. You got single-strand breaks, you got double-strand breaks. When they get repaired, they sometimes get repaired with not the same coding system that was there before, and you get a mutation in the gene. And sometimes those mutations make the gene not functional. Or if you have double-strand breaks, you could lose the entire gene. It could just be clipped out and it's gone. So that's what's happening, just like when you, illustrated on the right, is what happens when you go out in the sun and don't get in the sun without sun protection, because that's what you're getting, thymidine dimers, and of course, eventually you can get skin cancer from that. So these mutations are being accelerated in the children that are being treated with neuroblastoma for the chemotherapy. So if you look in this illustration, you start out with lots of tumor cells loaded with GD2, and illustrated therein is red is one that either de novo, and sometimes this happens, that de novo there's just low GD2 on that tumor cell, or more commonly, you've got this chemotherapy and it's causing this mutations, and you get a cell that has low GD2. And you're treating these patients with chemotherapy mixed with an antibody GD2, which is what we're doing right now in our national phase 3 study in the COG. And then as you go through that, you see illustrated is you're going to get an increase in the population of GD2 low, because they're not getting killed by the antibody anymore. That antibody is really benefiting. The immunotherapy is working, but it's not working if the antigen's not there. And then you get, and here's what concerns us, you get to the point where, where do we know anti-GD2 therapy works the best? Is maintenance therapy or post-consolidation therapy. When we've gotten rid of everything else, then we come in with immunotherapy on very small amounts of tumor. You saw that in just this presentation we just saw. She showed you that the patients that have high tumor burden weren't getting very much response. It's the patients that have low tumor burden and you come in with immunotherapy that it works. You get to that point in therapy and you've selected for tumor cells that have lost GD2. Our concern is that again, this is all theoretical at this point, but our concern is that that is where we're not going to see an impact of the antibody anymore. So it's a two-edged sword. You move antibody into chemotherapy up front, it's helping us. It really clearly is going to help. But is it going to increase the potential for resistance so the antibody won't work out the back end? And that's the chief concern we have right now. So does this happen? Well, if we take a bunch of cells growing in culture, cell lines, from patients, and this is each one of those is a different patient there that we've grown in the Children's Oncology Group repository for growing these cell lines from patients. And you can see in the light blue, those are patients' tumor cells that were grown in culture before any therapy. In the dark blue are ones that were grown at the time of relapse. And if we look at GD2 expression on the top bar, you see the percentage of the tumor cells that are binding. And as we all know from plenty of data in the literature, most neuroblastoma cells, a lot of them express GD2, they bind, you see that right there. And it's a mixture between the pre-therapy and the post-therapy of the low ones, but there are you see there, you actually see one pre-therapy one in the ones that have low percentage of positivity. But if you look at that, it's starting to tell you what the picture we're going to see. Most of those that have low therapy are ones that have already gone through chemotherapy, that have low GD2. So the selective pressure is having its impact. And concerning to us is the amount of antigen on the surface, which is that lower graph. It's not just the percentage of ones that we say, hey, they're positive for GD2, but the amount of that antigen on the surface to target is getting very low in these. So from these non-clinical data, we then asked the question, and that's Paul Sondel. Paul Sondel, you and I sat down at a Chicago airport here years ago, and the COG said, hey, we've seen your isotretinoin data, and we know that POG is about wants to work with the CCG to do an antibody study to prove that antibody therapy works. And we designed the study in this airport over here that day because it was, they didn't give us a choice. Come here to the airport, design the study because we're going to change it. We're going to add antibody together with Accutane. And so Paul and I have collaborated for many years, and I sat down with Paul one day and I said, how can we study this in mice? And he said, we looked at each other and he said, well, mice have NK cells. I mean, nude mice. Nude mice will grow tumor cells, okay? You know, we call them nude mice, they have no hair. They don't have a good immune system, so we can stick a human tumor cell in there and it'll grow. They don't have the T cell arm, but they got NK cells, they've got monocytes, they've got plenty of neutrophils. So well, this should work, but when we put antibody just by itself in there, it doesn't work very well. Of course, you've got a big tumor in a patient, you can give them antibody. Just by antibody by itself doesn't really work very well either. But we went in and we tried this with chemotherapy. And we said, well, just like Modi's study where he's combining it with chemotherapy, and we just saw that great activity. And sure enough, we could replicate that. You see there the control tumors on the left there. You can see these guys are growing very well. Antibody by itself not doing very much. And you give temozolomide or irinotecan. This is a patient-derived xenograft. Now, we took the tumor from a patient before they had any therapy, and we put them right into the mice and grew them and did this. And you can see we respond to the chemotherapy, temozolomide and irinotecan in this case. Well, look how much better the response is when we add the antibody on top of it. Just exactly what was seen in the clinical trial. And you can see the event-free survival. The mice there is extremely good. And on the lower right-hand corner, you can see the histogram. This is a flow cytometry diagram. Flow cytometry simply means that we took a whole bunch of the cells and we spit them past the laser at 100 cells per second, and we've put a fluorescently tagged dinutuximab on it so that every cell that has GD2 is lighting up with fluorescence, and then we're just counting those and drawing a histogram. You see that big right shift from the control is the gray. The red shows you that virtually all these cells have a lot of GD2 on the surface. So now we had a model to study this. And then we went into a variety of different patient-derived xenografts. And look from the right, there's your 480X for the comparison I just showed you. Go over from the right to the left, 452X. Here's a patient-derived xenograft from a patient who has gone through a lot of chemotherapy and had antibody therapy. They still have a fairly good amount of GD2 on them. They're responding pretty well, but look, not as well. There's lower amounts of GD2 and they're not responding quite as well. Now look at Felix, named for the child that I almost say studied. This is an index child on the crizotinib study. And when he died, we did a postmortem establishment of this patient-derived xenografts. The parents get to name the postmortems. And that's how that one got the name Felix. Very cute kid, by the way. We've got the picture up in the lab. And as a result of that study with that IL did, that's why lorlatinib is now being used instead of crizotinib. It was a fantastic piece of work that she did. But when we looked at this one, we didn't see very low GD2. It's still positive somewhat. But look what happens when we add that on top of chemotherapy. It doesn't do anything. 519X, another patient-derived xenograft from progressive disease that was virtually negative for GD2, and again, no response in these mice. So this concerned us a great deal, and that we might be losing the ability for these cells to respond to the GD2. So what we did then is we developed this assay. And in the days when we were having to purge bone marrow, a couple of you in here will probably remember those days. Most of you newer ones won't, but that's before we collected peripheral blood stem cells for transplantation. We used to have to harvest the bone marrow. It was loaded with tumors, so we had to get rid of it. So we used magnetic beads attached to a whole bunch of different monoclonal antibodies. And one of those was this antibody, HSAN. And that antibody we knew bound to all neuroblastoma cells we could ever find. It doesn't bind to GD2. At the time when we were doing this, we didn't know what the antigen was. We now know what the antigen is, an interesting antigen. But we knew it was very specific for neuroblastoma, so we pulled it out of the freezer and started working with it. And we said, if we light up, the problem is, as everyone in this room knows, it's just not feasible when we got to go in and biopsy a kid's solid tumor every few months, see what's going on. But it's really easy to get a bone marrow aspirate. We get them all the time. Now, bone marrow aspirate's a mixture of all kinds of cells, but we need to just find the neuroblastoma cells in there. So on this flow cytometer, we run through there, and the first thing we do, you see in blue, we light up all the hematopoietic cells, all the blood cells, and we just tell the computer, ignore those. And then next we go in and we light up with another color of fluorescence all of these HSAN positive cells, all the neuroblastoma cells. But that's all neuroblastoma is lit up by there, and then we come in with a third color, and we say, this is GD2. So we have dinutuximab conjugated with a red fluorescent dye that lights up all those, and then we look at that, and you see the histograms. We have bone marrows from patients shown there. You've got very high ones on the left, and on the right, you see one that's very low. So then the Children's Oncology Group was sending us a large number of samples from patients that are on a biology study. And we started looking at those. Again, this is giving you another look at instead of the histogram, we're now showing what we call a cytogram. So on that x-axis, on the grams on the left, that's the amount of HSAN. This is every one of those is a tumor cell, okay, because we've gated out the hematopoietic cells. And so there's thousands of tumor cells there, and you see the ones up in the up above the line there, they're all tumor, they're all neuroblastoma because they're HSAN positive. And all the ones over in the right quadrant, they're all HSAN positive and GD2 positive. And that's a typical diagnosis marrow. But look at this typical relapse marrow. Plenty of tumor there, but no GD2 spot at all. So then we looked at a large number of patients here, and this is 93 patients that had bone marrow, and then a few that we had tumor and blood on with this assay. And it shows you that we can do it across any of them, but of course the most useful specimens for us are going to be bone marrow, because that's most of what we got. And on the right shows you bone marrow comparing the patients at diagnosis, and this is not the same patients, this is different patients, but another group of patients that progressive disease. And there's a significantly lower amount of GD2 in those and most of these patients have been treated with antibody therapy. So that's the thing that's concerning us, is that the loss of this. And when we saw in the mice, well, does this mean anything? Well, the bottom line is, at this point, we don't know. But that's being studied, okay? So before we talk about what we're doing, we'll just review this summary here. That clearly the enhanced killing of neuroblastoma cells by adding anti-GD2 therapy is a very important way to enhance chemotherapy. We see this in patients, and we see this in our preclinical studies. But it requires, as we see in the non-clinical studies, you've got to have GD2 on the surface. Neuroblastoma cells in some patients can have very low amounts of them, so it's possible that they won't respond to anti-GD2 therapy, no matter what you use for anti-GD2 therapy. We don't know that, but that's what's being studied. It's a complex molecule. It's 9 steps in it, so the chances to lose it are high. And combined DNA damaging agents, of course, is mutagenic, so that means you could clip one of those genes. So that's really the concern with the combination with chemotherapy. And we have clear data now that neuroblastoma patients, at least some of them, have low GD2. So in the Children's Oncology Group, we have looked at this in the most recently completed phase 2 study, which is the DFMO plus anti-GD2 plus chemotherapy versus antibody plus chemotherapy alone. We don't have the response data yet, but we have analyzed all those for GD2s. Smaller number of patients, but we can get something out of that. I think we'll learn something. But the most important data is right now is that in the Children's Oncology Group is right now, Sarah Federico out of St. Jude's leading this study with Tom Cash, taking the antibody and moving it into upfront chemotherapy. And of course, they'll also get the same antibody out the back end. So we're randomizing patients to get antibody plus chemotherapy versus chemotherapy alone like we usually do. And in every one of those patients at diagnosis, and then any point along where we're getting a bone marrow, and particularly at relapse, they're getting cells into our laboratory and we're analyzing them for GD2. So at the completion of this study, we will understand really what this means. I think we're going to have to wait for that to understand it, but I think it's going to be very definitive. Out of the first 12 patients on the study, 4 had to get treated so quickly that we couldn't get a sample. There weren't one sent, but 8 of them sent samples. They all got analyzed. We got 3 cell lines growing out of those, so we can go back and replicate this in the laboratory. So we're very happy how this study is going right now. And I'm pleased to say that Dr. Seibel sent me an email last night that said that the NCI is going to fund the study. That was not approved until yesterday, so that was really good news. So I think we'll learn a lot from this. I know that in a lot of your questions are saying, well, what do we do with our kids? What can we do with this? We don't know the answer right now with this. This is not a clinical test. We can't look at somebody and say, you shouldn't get GD2 or you should get GD2 therapy. But I hope I convince you it's very important that we look at this every chance we get. And in fact, the NANT, Araz is back there, and they're looking at it with us in their studies as well. All right, I think it's in any of the core COG studies are incorporating it. So I think we'll learn a lot from that. And the next time we talk about this in a few years, we'll be able to say what it means. I wish I could tell you right now we had a clinical test, but it is clearly an important test to study. And I'd like to acknowledge Michelle Kyle, the PhD immunologist in my lab, this postdoc that's done a lot of this work. She got the only award to a U.S. person at the A&R meeting last year for this work. So we're very proud of that. She got one of the 3 awards there. And Cezanne is the MD PhD student who is working on this with us as well. They've done a great job. And of course, the support from the NCI, the Cancer Prevention Research and Infectious, and Alex Lemonade Stand Foundation has helped with this. Julie Park, Arlene Naranjo, and Ro Bagatelle, Meredith Irwin, Mike Hogarty, and Paul Sandell, all in the COG. They couldn't do this work without them. And of course, Nita Seibel at the NCI. But most importantly, the samples that come into our lab come because parents and children are willing to let us study those samples, and we're very appreciative. So I'd be happy to take any questions.
Dr. Reynolds. Anyone have questions?
Thank you for that presentation. And this perhaps is very naive. I'm sort of new to all of this biochemistry of neuroblastomas, but is there any way in which you could enhance the GD2? I understand that.
That's a great question. You may be new to biochemistry, you're not new to neuroblastoma. I know that for a fact.
No, I'm not.
There is papers in the literature, and there's also people studying this. In fact, one of the projects in the NANT program project that was recently funded is looking at just this, and we're looking at it as well. Looking at drugs that might enhance the expression of GD2. Mm-hmm. I can tell you there are going to be some where that won't work. We know already because of whatever's happened. It's just not enhanceable. But that's certainly something that a number of laboratories are looking at. Cezanne, who I showed you the picture of, that's his MD PhD project, is really mapping out each of the mechanisms of all of the tumor cells we find that have low GD2, and they differ. It's not just one size fits all. There's a lot of complexity there. But it's certainly a very promising area. And in fact, there's some xenograft studies going on right now at CHOP, I know, looking at this, that we're hoping are going to work very well.
Is there any particular chemotherapeutic agent that seems to be more destructive of that GD2 than others?
No, I think, any cytotoxic therapy is got the potential for enhancing mutagenesis.
Okay.
And importantly, we know from our data as well as the Germans, it's not just that. If there are some patients neuroblastoma patients that just don't have GD2 to start.
Oh, okay.
Fortunately, it's a low number, but there are some. So I don't think there's any chemotherapy I can think of that would be less or more likely to do this. They all hit DNA.
Okay. Thank you.
One question there.
So you use the HSAN as a biomarker. Is that a potential for creating an immunotherapy or kind of like a combo of GD2 plus the HSAN? I don't know what else it's expressed on.
Well, that antibody I created when I was a medical student, okay, and I had it a long time. But we had no idea what it bound to. In a Stand Up to Cancer grant, John Maris was chasing the best immunotherapy targets that he could find, okay? And he identified GFRA2 as one of the primary targets they wanted to go after. They were trying to make antibodies to have trouble. Well, Michelle actually showed that HSAN binds to GFRA2. So we sequenced that antibody. We sent it to John Maris. His collaborators made a humanized version, which we're studying right now. So we're very much looking forward to trying to see if that will work in the GD2 lows. So it's a great
definitely.
question. We don't know yet, but
Because you had the population that was depleted in GD2 but still had the HSAN.
yeah, the first of those experiments is going on in the mice right now.
Okay. Awesome. Thank you.
Any other questions? Okay, we got another one. Go ahead.
Sorry, long walk. You mentioned, well, I have two questions. So first, you mentioned that you did these flow cytometry things. Is that available for our kids? Are there like liquid biopsies? I don't know if that's the right term for this, that we could just see if they have high levels of GD2 expression?
Well, it's a research test at this point, so we can't use it to guide therapy. Okay. So we have to prove what it means. And then once we do that, we can get it into clinical labs that make it a routine test, but we're not quite there yet.
Got it.
I mean, I would encourage you to have your patients enroll, any children you have enrolled on COG studies or NANS studies because it will get done. But we can't really guide therapy at this point till we get more data and understand exactly what it means.
Okay. And then what happens, or let me rephrase this, what options are there for kids who lose GD2 expression and they're in relapse? It does seem like most of the treatments just target GD2, like the CAR T, and as you said, the Dynatuximab.
Well, there's other antibodies already in development. I mean, I mentioned one that we're looking at HSAN. It's very early, but the GPC2 is well along the way. There's a couple of them that people have in CAR Ts or as trying to get antibody drug conjugates that are to other targets. They're very early phase trials, so it's nothing that's late at this point. But there's certainly a lot of work going on to find other targets.
Okay, thanks.
Thank you. Anyone else? Anyone? Okay. Thank you. Oh, one more? Wait, we got one more. He's over on that side.
So, let's see. My question is, our son, MIBG, went from being able to see on the scans to not. So would the GD2 have anything to do with that and like immunotherapy not really working very well for him? Does that make sense? Like, is that a marker that could tell that the genes have been changed where the GD2 wouldn't be present anymore?
If somebody's getting immunotherapy with anti-GD2 and it's not working, this is certainly a possibility.
Okay.
It's not. We don't have the test at the point where it can be used to say, "Hey, you should not get this therapy," or "That's why it's not working." But in a research context, I think it's worth looking at those questions. And any studies, a lot of studies are doing that now, but it's not something that's just routinely available yet.
Oh. And then is the GD2 how's it measured?
How's it measured?
Yeah.
Dinutuximab is the antibody that we most commonly use in this country to treat with as an antibody that binds to GD2. We have it labeled with a fluorescence molecule, and then any tumor cell that has GD2 will bind the dinutuximab, and that will cause it to glow. And that's how we measure that in that instrument called a flow cytometer.
Okay.
One more question?
Yeah, I'm curious if the chemo is the thing that's causing the mutation, and I know that our kids get a lot of chemo, but then, especially up front, oftentimes with relapses, we do chemo and immunotherapy, not just chemo. And I guess maybe I'm speaking more from my own experience. How do we know when to drop the chemo and do just immunotherapy if there's no evidence of disease? You know how if you're doing chemo and immunotherapy, you gain no evidence of disease. How do we know what's most beneficial, to drop the chemo or to keep it with the immunotherapy if the chemo's the thing causing these mutations?
Well, it's a good question, okay? I mean, within the COG, as everyone knows, our approach to immunotherapy early on was to say, let's not get into this. Let's put it out the back end, okay? So we're not mixing it with chemotherapy. Then came along responses to patients with relapse disease with chemoimmunotherapy. So now we're moving it up front. I think we have to look, as we're doing in this current, the ANLB 2131, this large COG study, what's happening on various time points on all these patients to their GD2 to understand and answer your question. We don't know the answer at this point.
Okay, I understand that. I know there's a lot of unknowns. What is it about the chemotherapy that makes the immunotherapy work better?
That's also unknown, but I'll tell you what I think it is, okay? All right? As best we can tell, what happens is when you start using chemotherapy, you're killing some of the tumor cells. That's causing infiltrations of monocytes and neutrophils because you've got a lot of bad things going on in that little microenvironment. They get in there, and those cells come in there, and they come in angry. You arm them with an antibody, and they get the tumor. But that's also something that's not well understood at this point, but that's what we think is going on.
Okay, thank you.
Sure.
Thank you. One more question? Yes?
So the COG protocol is typically 6 rounds of immuno with the DINA. Sometimes they recommend to do additional immunotherapy if a stage had to been cut off. So I ask if it gets a little easier each round with the DINA, like less of the reactions and, they're less pain and all that. Does that mean that there's less of that molecule that the DINA is attaching to?
No, any side effects from the antibody have absolutely nothing to do with what's going on in the tumor cells.
Okay, cool.
Okay. So, and again, if there's one thing I want to stress right now, we don't know yet which patients would benefit or not benefit because of GD2 levels on their tumor cells. So the safe thing to do is treat them with the antibody at this point, or whatever immunotherapy is going on. But again, when we're doing clinical studies on that, it's really important also to look at this question so that we can give you that answer in the future.
I think that's
Thanks,
it. Thank you, Dr. Reynolds.